Welcome back to part 3 of Unit 3, Lesson 2. This is optional unless, of course, you've missed class. So we were talking about how topoisomerase unwinds the helix, helicase unzips it. Now we're actually ready to start replicating it. So in step 3, we have our DNA strand that's been unwound and replicated, unwound and unzipped. And we had over here the ATGC, and on that side TACG. And up here, working ahead, was topoisomerase to unwind it and helicase to unzip it. And now what's going to happen is in comes an enzyme called DNA polymerase. Okay. If we make this the 5' side, and this was the 3' side, then what's this have to be because it's anti-parallel? That's correct, 3 to 5. It's going to start building an identical copy, or actually a complementary copy to each side of the chain, but it has to build in the 5', 3' direction. So polymerase is going to come in and build in this direction, 5' to 3' on that side, whereas on this side it's going to come in and build in the opposite direction. And it's going to do its job. What it does is it helps bring in the base pairs and build or complement the base pair. So it would have started down here, bring in a G, then in would come a C, in would become an A, and in would come a T. Over on this side, would have brought in an A, then a T, and then a G, and it hasn't got down here yet, so we won't show that anything's happened yet. And it builds the new strand, complementary. If you look, if you're on this strand, it can just follow up right behind topoisomerase and helicase, no problem. But that's called the leading strand, because it just follows along. It actually gets a head start on the other strand, which is called the lagging strand, because the other strand has to wait for topoisomerase and helicase to get out of its way before it can start building down below it. And it actually ends up building in little chunks. And these little chunks need to be glued back together at the end. And that's done by DNA ligase. So let's just write that all out, because we've sort of gone ahead of ourselves. So in step 3, what's called DNA polymerase, often just called DNA pol. Think about the name polymerase. It's making a polymer of DNA. It's bringing together the nucleotides and building a chain. So DNA polymerase brings in and attaches together complementary base pairs to build a chain or a polymer of DNA. But when you're building that chain, sometimes you get little nicks in the backbone, the sugar phosphate backbone, in particular in the lagging strand because you have to build them in little chunks. So you also have an enzyme called DNA ligase. Ligase is a fancy word for glue together. Ligase glues together any nicks in the DNA sugar phosphate backbone. And this will go on and the entire length of the chromosome until you get 2 complete new copies, which are attached together in the middle by the centromere, and that's when you end up with 2 identical sister chromatids. So in our final picture, we show that we have created 2 daughter strands which are identical to the parent strand. I'm just showing that the new strand it results in 2 daughter strands, barring the mutation, identical to each other and original parent double helix. I just want to point out there's a special term here. It's called semi-conservative replication. Semi means partly or partially, and conservative means to keep the same. And the reason it said it's semi-conservative is because each daughter has 1 parent strand, so 1 strand from the original parent, and 1 newly made strand. And this is how we're able to build generation after generation identical DNA strands based on complementary base pairing to the original parent strand. And that's it. That's how replication occurs, and then the cycle can continue onward over and over again as needed by the cells for division. Okay? So just remember, it might be a good idea at this point, your key players here, your key enzymes, there's 5 of them. No, 1, 2, 4 of them, sorry, are topoisomerase that unwinds, helicase that unzips, DNA polymerase that actually builds the chains, and then DNA ligase that will glue together any nicks in the sugar phosphate backbone. Okay. If you have any questions about that, you can come see me in class. Otherwise, I hope you have a good day. See you in class.